much. And we'll start with our first speaker, Dr. Grace Lee. Dr. Lee, as many of you may know, is an assistant professor in endocrine surgery. She received her medical degree from the University of Oklahoma College of Medicine, did her clinical training, residency, and research fellowship at the Brigham and Women's Hospital at Harvard Medical School, and then did her fellowship in endocrine surgery at the Mayo Clinic before we were very fortunate, obviously, to recruit her here to Yale. Dr. Lee is really an innovative expert in endocrine surgery and is looking at new ways to combat, obviously, thyroid, parathyroid cancer, among others, and seeing how we can, leverage new techniques to improve the care of patients and have a firmer understanding of the disease. And Grace, I want to thank you for taking the time to join us for Kansas City Grand Rounds, and welcome hearing you talk.
Thank you, Dr. Fuchs. That was a very kind introduction. I am delighted to be able to present an area of my clinical interest with everyone. I can share my screen.
It works.
All right. So my name is Grace Lee. I am a new-ish endocrine surgeon to Yale. I was very fortunate to join Yale earlier this year, sort of at the height of the pandemic, from Tulane Medical Center down in New Orleans. And I have been an early adapter of this new technique called radiofrequency ablation in thyroid at Tulane. So I just wanted to share my experience and what I know about radiofrequency ablation. So hopefully we can also start this treatment at Yale as well. I have no disclosures. My talk will briefly touch on several different areas. I'll just touch on the background of how the RFA works, the physics and mechanism, and brief history. Current procedural guidelines. When we are deploying something new, there are governing bodies who guide us as far as indications and when not to do it are concerned, and we'll cover why we should be looking into this, what are the risks and the advantages. And we'll also visit efficacy and safety of this technique, as well as I'll show you a little video clip of how this is done and future directions. So percutaneous ablation is a technique that obviates the need for a surgery, and I'm sure many of you are familiar with these needle, often imaging guided way of eliminating undesirable tissue. There are multiple different ways of burning or zapping things. We have utilized laser, microwave, cryotherapy, freezing it, burning it, even injecting ethanol to chemically sclerose things, or HIFU, which is high intensity focused ultrasound. Today, we are going to focus on radiofrequency ablation, which is on the spectrum of these energy harnessing ablation techniques. Radiofrequency, just a brief physics, is concentrating heat or thermal energy at the tip of an electrode catheter by alternating current. And this frictional heat that gets generated from ionic molecules will turn into conductive heat from this frictional heat and create this ablative unit. And if you sustain this heat enough in that area, this thermal energy will cause irreversible cellular damage, so essentially coagulation, necrosis, but without charring the tissue or carbonation of tissue. Radiofrequency ablation in medicine, interestingly, has been utilized for a long, long time. It has been around in medicine for over 75 years. And some of the areas that have already started using radiofrequency ablation are wide. Cardiology started using this for arrhythmia, vascular surgery, surgeons use for varicose veins, orthopedic surgeons, and for us, there are oncologic purposes for radiofrequency ablation that you may be familiar with for either metastatic or primary liver lesions in pulmonary mets, adrenal mets, solitary renal lesions, as well as Barrett's esophagus. History of thyroid RFA is not as deep as the history of RFA in other parts of the body. 2001 was kind of burst, here I am year for thyroid RFA. The paper at the top, Percutaneous Radiofrequency Ablation of the Thyroid Guided Ultrasound, is a single animal report. The animal used was a pig in Japan. And this was just a proof of concept. Is radiofrequency ablation feasible? Is this safe? And subsequently, it's interesting that our colleagues at Brown, both interventional radiology and surgery department, got together and tried this RFA in recurrent thyroid tumors. So this technique has been utilized, has been kind of played with for now nearly 2 decades. The person who actually took it and ran with it is this physician interventional radiologist, Dr. Beck. Dr. Beck is located in Asa Medical Center in Seoul, Korea. He himself also had multiple benign thyroid nodules, and about 25 years ago, he was recommended by Korean surgeons to undergo a thyroid surgery. And he did not want a surgery, so he started looking into what other ways are there to eliminate thyroid nodule, and he started to focus on RFA. Not only he started doing this for patients, he himself has undergone this treatment. So he has been really transparent about the results of RFA, and he has been a passionate advocate for how this technique should be utilized and efficiently, safely. And he has given multiple talks. He's been the most prolific writer in the RFA literature for this technique. So in 2009, the Korean Society of Thyroid Radiology came up with recommendations for how thyroid RFA should be deployed. And in 2012, they came up with the first edition of guidelines for thyroid RFA. In 2017, they revised the guidelines and came up with a more updated version of thyroid RFA guidelines. Since then, this technique has been utilized worldwide, and multiple hands-on teaching sessions and workshops have been taught by Dr. Beck and others. So Japan, Taiwan, obviously a lot of Asian countries, as well as European countries have embraced this technique, and it has slowly gotten to the US as well. So last year, this is obviously pre-COVID medical conference that we don't have anymore, group picture, folks from all over the world came together for this Korean Society of Ultrasound and Medicine conference, KSUM in Seoul. And I was fortunate to participate in this conference and learn RFA from this conference as well as Dr. Beck afterwards. So there's a great interest in the things that I have learned, and the collaboration and the friendships that I have made in Korea have been invaluable. And it really has helped me a lot in deploying this technique at my prior institution at Tulane. So in 2015, Italian radiologists and endocrinologists came up with a consensus opinion. And in the UK, they came together and consolidated a recommendation in 2016 and 2017 in Australia. So what about us? What about the US? Why are we not widely adopting RFA? Why are we just now catching on? So there are multiple reasons as to why the US sort of has lagged behind in adaptation of this technique. It's not that clinicians in the US have not heard of it, it's just that we have been a little bit more cautious. And as of late, just in the past couple years, there has been a booming interest in RFA technique by multiple societies who deal with thyroid pathology. And I'm sure that this RFA technique will be widely deployed and widely utilized. And I hope that Yale will also start offering this treatment as well soon. So the seminal event that kind of took off the interest in the US, I believe, is this paper, which came out of Mayo Clinic in 2018. The group here are comprised of endocrinologists who actually do their own FNAs and utilize ultrasound in clinic to assess thyroid nodules, as well as prominent interventional radiologists. They have published this case series comprising 14 patients over the years of 2013 and 2016. And their result really has been terrific. They achieved volume reduction by 45% as early as 8 months and durably at a couple of years out, and very safely. So I think this paper actually sort of put RFA on the map for the US clinicians and audience. So since then, there has been many workshops and conferences, and as of late, I have been getting presentation notices almost monthly. So why are we excited about RFA? Like, what are the advantages? So one advantage that I, as a surgeon, really appreciate is that patients have lower anxiety about undergoing a percutaneous, minimally invasive treatment rather than undergoing a surgery. Most of my patients, unlike patients in Korea, are a little bit less concerned about the cosmetic outcome, the scar after surgery, but they are still very anxious about just the prospect of having to undergo a surgery. So I think this technique, if the patient qualifies for it, will be highly beneficial from reducing anxiety perspective. And obviously this is percutaneous needle poke. There won't be a long scar that will be left behind. Of course, in thyroid treatment world, there has been a couple of modalities where we are taking out thyroid through the armpit, axilla incision, through the mouth, transoral method, but each one of those scarless or scar hiding surgeries come with either an increased cost of robot deployment or longer surgery, longer recovery, etc. So those are, they probably have a role in the world of thyroid treatment, but they're still not as optimal if the patient is truly aiming for the best cosmetic outcome. And it's a needle poke, minimally invasive. There's no hospital admission. Some of the thyroid surgeries that we do, of course, are outpatient, but there is still recovery involved after general anesthesia. There is pain involved. But with this percutaneous technique of RFA, there's no general anesthesia. There's only one institution in the US currently who's doing this RFA under general anesthesia. In Korea, this RFA is actually done in a clinic setting. with no max sedation, just under local anesthesia. It's a relatively short procedure. Obviously, is volume of the thyroid nodule dependent. Bigger the nodule, longer the procedure, but it's a relatively short procedure. I would say anywhere from 10 minutes to 30 minutes per nodule. And repeat ablation is a possibility. So you're not burning any bridges if the first go does not work out for the patient. No no hypothyroidism, which is remarkable because we're only ablating the nodule and preserving the normal parenchyma of the thyroid. Is virtually unheard of to get hypothyroidism after RFA treatment. And also because it's nodule specific, there's no hypoparathyroidism either. So minimal complication risk. There could be voice changes and temporary discomfort, but compared to surgery, I would say it's either equivalent or less, and overall lower cost. And that comes from not admitting the patient, no prolonged OR time or anesthesia, etc. So what are the downsides to RFA? The risks, there are several. I just mentioned that repeat procedure is a possibility, but sometimes it's a necessity. So just one go around may not be enough to achieve therapeutic effect. So obviously having to do a repeat procedure is a risk. And I would also say that it's operator dependent. Many procedures are operator dependent, surgeries are operator dependent, and that's why we advocate for high volume endocrine surgeons to be doing endocrine procedures. Just the same with RFA. The more facile the operator is with ultrasound and needle guidance, the better the outcome will be. And some patients may not be able to tolerate a procedure without sedation, whether it's related to pain or anxiety. It's quite interesting to see the stoic nature of these Korean patients that I observed at Asan Medical Center last year. I know that 18 gauge needle that's almost looking like a harpoon hurts, but they were very stoic. They said that they're not having any pain and they tolerated just fine. But when I have been doing fine needle aspiration thyroid nodule biopsies in clinic setting, although it's 27 gauge needle, much finer, many of the patients complained of pain. So it will be interesting to see how many patients can actually tolerate this kind of a procedure without sedation. Complications. When there is something being done to any part of the body, there could be associated complications. Overall rate is pretty small at 2 to 3%, which is probably equivalent to a surgical treatment. There could be dysphonia, which is about 1%, and this data is coming from multi-institutional pooled data. So about 1%, and none of it was permanent. It was all temporary, resolved after 2-3 months. Hematoma, thyroid is very vascular. Skin burn, that's probably a little bit more prevalent in RFA setting rather than surgery. There are a couple of lower incident complications such as tumor rupture, which is unheard of in surgery, hypothyroidism, abscess, and a couple of anecdotal, not reported in this study, complications such as tracheal injury and brachial plexus injury. When a new technique is being deployed and multiple people are just trying it out, I think that there will be a wide variety of complications that may arise as well as the outcome. So RFA and thyroid, there are guidelines and indications, and most of it is based on 2017 revised Korean guideline. So these are indications that, that most experts agree that we should be doing this for benign thyroid nodules causing compressive symptoms, if the patient has cosmetic concern, and the nodule should be of size, at least 2 centimeters or so. and if it's enlarging but has benign cytology, but we have not set any definitive maximum thyroid nodule size for the treatment. And recurrent cyst after failure of ethanol ablation, experts will agree that ethanol ablation is more studied, more tried, and better method for recurrent cyst or cyst of the thyroid treatment rather than RFA. Autonomously functioning thyroid nodule, this is toxic nodule indication. I personally have tried RFA in toxic nodule. It works beautifully, and that sort of made me a believer of RFA technique. Entirety of nodule should be visible on ultrasound for safety concerns. It should not have any substernal component. And nodule should be sufficiently away from the vital structures in the neck. As you all know, there are nerves and vital blood vessels in the neck, and since the heat can transmit, it is important that the structures are constantly being observed and monitored so we don't cause any undesirable damages. RFA is most effective in solid nodules, and if the nodule contains macrocalcifications, the RFA will not be as effective. Ideal candidate, the pathologic criteria, we want to be 100% sure that it is benign by either 2 repeated ultrasound guided FNAs. And in Korea, what they do is core needle biopsy. This is not a universally practiced way of establishing benign status of the thyroid nodule, but core needle biopsy is an alternative to fine needle aspiration, and we want to make sure that there isn't any sonographic features of malignancy seen prior to deploying RFA. And again, we want to make sure that surrounding critical structures are away from harm. RFA guidelines, so multiple different countries are coming up with guiding this RFA treatment to be deployed. Obviously, Korean guideline is the most comprehensive. Italians, Australians, AACE, American Association of Clinical Endocrinologists, we have a little bit of more conservative recommendations for RFA. So we are currently only recommending RFA to be used for benign thyroid nodules that are causing cosmetic concerns or symptoms and should be biopsy proven to be benign. Contraindications, who cannot undergo this procedure? Pregnant females, if the patient has contralateral vocal cord palsy, if the nodule is not seen in its entirety, the patient has implanted cardiac device, we can switch over to bipolar mode instead of regular monopolar electrode, or deploy it in shorter bursts and lower energy. Or if the patient has anticoagulation on board, it should be stopped prior to RFA. The indeterminate thyroid nodule is a gray area. There has been a study published in Europe, but it's just a limited number of patients, and the efficacy and the long-term outcome of doing RFA in indeterminate thyroid nodule has not been quite established. And malignancy, we are pushing a little bit of the boundary on this malignancy territory. So I'll discuss that briefly since we are under the umbrella of Smilo Cancer Center. RFA and thyroid, when we're dealing with toxic nodules, works beautifully. If we can achieve at least 50 to 80% volume reduction, TSH normalizes, and by 12 months, most people are euthyroid. But again, if your desire is to achieve immediate reversal of thyrotoxicosis or hyperthyroidism, then RFA is not the way to go. And it may take more than one ablation session depending on the size, and oftentimes these toxic nodules tend to be a little bit on the larger side, greater than 4 centimeter. So in that case, combination of RFA and RAI can be entertained for large toxic goiters. This is how the electrode of RFA works, and how the picture shows you how fine it is. This is much finer than liver RFA electrode, and this just purely equipment optimization is what has taken the RFA deployment in thyroid lag behind compared to the liver world. So this electrode is internally cooled. It's about 18 gauge big and 7 centimeter in length. So if we're dealing with a very big patient, that comes into play. So liver has deployment of tines, whereas RFA and thyroid, we are actually introducing the electrode and pulling it out, which is called the moving shot technique, to ablate units, these circles of ablation zone inside the thyroid nodule. And you see that a triangle containing recurrent laryngeal nerve and esophagus below. So those are the vital structures that we want to be avoiding. This is a brief video of mixed thyroid nodule that is undergoing radiofrequency ablation. You see from the isthmic side, introduction of the electrode. We want to be able to see the entire length of the catheter, and it is introduced and treated, ablated from posterior to anterior and inferior to superior. So we actually ablate the entire ellipsoid thyroid nodule. Follow-up, we do serial ultrasound to make sure there's an adequate resolution of whatever the pathology that is being treated. And there is a concern for regrowth, and that is partially due to inadequate ablation at the periphery. So complete ablation of the thyroid nodule is being advocated. I'll touch briefly on the thyroid malignancy and RFA. Currently in the US, not many centers are doing RFA for thyroid malignancy. However, it has been done in other countries, and more folks are pushing the envelope, especially in the world of micropapillary thyroid carcinoma, which is less than 1 centimeter. If the micropapillary thyroid carcinoma is intrathyroidal and there's no clinical or ultrasound evidence of lymph node spread, the current American Thyroid Association guidelines say that you can either observe it with active surveillance or have the patient undergo a thyroid lobectomy. Well, over time, we have found that even the patients who undergo active surveillance, there is about 13% operation rates. So instead of having these patients undergo an operation, why not RFA? So clinicians overseas have published their data on micro-PTC, and the data is new and has garnered wider adaptation just yet, but I think it is coming. In recurrent thyroid cancer, as a surgeon, re-op neck for cancer is a bare territory. So if we can safely treat patients with percutaneous method, it will be fantastic. Indeterminate nodules, as I briefly mentioned, there's lack of data on whether we should be using this technique for Bethesda 3 and 4. So currently not recommended, and this is to be determined. Briefly, I just wanted to show you a little graph of how 5-year follow-up of 84 low-risk micropapillary thyroid carcinoma had achieved 100% regression over 5 years. And this is just showing that there's no neoplastic change secondary to radiofrequency ablation of thyroid nodule. It's mostly just a benign cellular change. And because RFA does not alter the thyroid capsule or cause neoplastic change, it is okay to redeploy RFA if needed or have the patient undergo a surgery if needed. So I'm going to just wrap up my talk by saying that RFA availability and indications will continue to evolve. And I think this is not a fad, but this is a treatment method that will probably stay. And management decision really should involve multidisciplinary team of experts and include the patient in the decision making. And I think this technique, given that it's relatively new to the US and may involve specialists from the interventional radiology, surgery, and endocrinology, and multiple hosts of pathology, et cetera, it is fitting for us to be introducing this technique as SMILO. And I am a strong proponent of inviting the patient into making what is best for them. So it is important for us clinicians to not introduce our own clinician bias. Just because I'm a surgeon, I'm not going to advocate a surgery. And this is not a technique that I currently subscribe to 100%, but I do believe that it has a role, and I'll do my best to be impartial and introduce this technique to Yale. So more research obviously is needed, and we're actively doing a project looking at our own institutional data. So I'm just going to wrap up by showing you this quote by Hippocrates. Those diseases that medicines do not cure are cured by the knife. Those that the knife does not cure are cured by fire. Those that fire does not cure must be incurable. But this philosopher also said, first, do no harm. So it is really a responsibility, and it's imperative that we deploy this technique safely. And I'll take any questions, and thank you for your attention.
Well, Grace, thank you. That was really a terrific summary, and it's really wonderful that you're bringing this technology and technique to bear for our patients at Yale. Just because we're running a little late, I'm gonna suggest that anyone who has questions should probably reach out to Grace directly, and turn now to our next speaker. And really, our next speaker needs no introduction. Dr. Haifan Lin, as you know, is the Eugene Higgins Professor of Cell Biology, Professor of Genetics, Obstetrics, Gynecology, and Reproductive Sciences, and he is the director of the Yale Stem Cell Center. His work since joining Yale in '06 to launch the center has really taken this center to national and international prominence. His own work focusing on self-renewing mechanisms of stem cells has really informed cell biology and cancer in multiple ways. And I think he's going to share with us the work that he's done in terms of a gene family that he and his lab have discovered and its relevance to cancer. So Haifan, thank you very much for speaking today.
Well, thank you much, Charlie, for a very kind introduction. I'm delighted to have this opportunity to give a progress report on my own, to my own colleagues at Yale Cancer Center. Although I've worked for many years on stem cells and developmental biology, regarding cancer research, I'm completely a new kid on the block. So in the past few years, our research on gene regulation of stem cells led us to the cancer field. So today, instead of talking about my usual type of stem cell research, I'm actually going to share with you some preliminary results of our recent cancer work. And this actually is my first time talking about these results. I hope to get your expert input, advice, and potential collaboration. So to begin, let me share my slides. Screen sharing. Okay, Can you see my slides?
Yes.
Okay, great, wonderful. Yeah. So, you know, as Charlie mentioned, I will first give a bit of background about some of our earlier work to pave the way for my cancer results. So in 2000, in actually 1998, we discovered the Argonaute gene family as the first gene family that's highly conserved during evolution for its stem cell function. And this gene family encodes a highly conserved group of proteins called the Argonaute proteins that contains 4 functional domains. Among them, the PAG domain and mid domain binds to small RNAs, whereas Piwi domain actually resembles RNSH. And this gene or protein family has been subdivided into 2 subfamilies. And you probably know very well about the Argonaute subfamily, which plays a central role in the microRNA and the RNAi mechanism. And these proteins directly bind to siRNAs and microRNAs, which are in general 21 nucleotides long. And because Argonaute subfamily proteins are expressed in most type of cells, that's why RNAi mechanism and microRNA regulation works for all of us who are doing this type of experiments. However, as a stem cell person, I was more interested in the other subfamily, the Piwi subfamily, because it's mostly expressed in the germline and in the most primitive stem cells. So in 2006, my lab and almost simultaneously 3 other labs discovered that the Piwi subfamily proteins bind to yet another class of non-small coding RNAs that we named Piwi interacting RNAs, or pRNA for short. So pRNAs are just like Piwi proteins. They are mostly expressed in the germline, and they are somewhat longer than microRNAs or siRNAs. So in this slide, I just want to share with you some salient features of pRNAs versus microRNAs, which are better known. In addition to being a bit longer, pRNAs are very complex. So in my lab, we today have cloned over 16 million of these pRNAs, which is in sharp contrast to the 1,000 species of microRNAs that's known in any organism. And in addition, the pRNAs target all types of genomic sequences, again, in sharp contrast to microRNAs, which mostly target the 3' UTR or mature messenger RNAs. And finally, the biogenesis is very different. pRNAs are mostly produced from long single-stranded RNAs, in contrast to microRNAs, which are from hairpin RNA precursors. So these discoveries are very interesting to us because, as you all know, that the molecular biology field relies on a very important principle called the central dogma, which tells us that genetic information in DNA is transcribed into messenger RNAs and then further translated into proteins. And that's how life's process starts. The discovery of microRNAs and small RNAs like siRNAs further enriched this dogma by telling us that now there are 2 new mechanisms regulating the stability or the translational efficiency of microRNAs. However, when we looked into where these genes encoding pRNAs reside in the genome, to our big surprise, they do not reside in this known gene coding region, which only represents 1 to 2% of our genome. Instead, they mostly reside in this junk DNA, what we used to call junk DNA, that represents a vast majority of our genome. And it is those genes that produce these enormous number of pRNAs. So we were euphoric about this discovery because if you liken our genome as a world, then the traditional genes are like the old world, the Asia-Euro continent. And somehow our research let us land ashore of a completely new world with pRNA genes and now many other genes. So this actually was a pie I baked for my lab to celebrate the discovery of pRNA. And it's my first baking product. And also this discovery was selected by Science as one of the 10 breakthroughs in 2006, the only biology breakthrough that was selected. So this excitement aside, the key question really is, does any of these pRNAs have any functions? So in past several years, my lab has been focusing on these pRNAs functions. It turned out these pRNAs and their partner Piwi proteins, they can be present sometimes in the nucleus and sometimes in the cytoplasm. So we wanted to first know when these Piwi pRNA complexes are in the nucleus, what's their function? To address that question, I'd like to bring your attention to a field called epigenetics. You probably know that epigenetics is a very exciting field that studies gene regulation. And the focus of current epigenetic studies focus on identifying epigenetic factors and illustrating how they modify the histone or DNA modification status, therefore turning on and off the expression of genes. However, to me, these epigenetic factors, they are so-called down molecules. They mostly don't bind to DNA, let alone recognize specific DNA sequences. But somehow they have to be guided precisely to specific genes in the genome, for example, here, but not here or here, in order for epigenetic programming to work and for the life process to unfold. So to me, a central question in epigenetics is, what are the mechanisms inside the nucleus that can guide these enzymes to the right sites in the genome at the right time in the right type of cells? When we started our work, a few epigenetic factors were illustrated to be guided by a few transcriptional factors to the promoters of a few genes. And that was exciting, but it's more an exception because first of all, it represents a feedback mechanism. And secondly, it was only accountable for a very small number of genes in our genome. So the big question to me existed and still in a way exists is that what's a mechanism that guides these epigenetic factors to their target sites? So fast forward to today, after several years of research, we discovered that the Piwi pRNA complex actually play a central role, at least in the germ cells, to guide epigenetic factors to their target sites. So this is how it works. So the Piwi protein and its partner pRNA will form a complex. And this complex will bind to genomic sequences with complementary sequences, actually by binding to a nascent RNA that's tethered still to the genome. This then leads to the recruitment of this complex to other epigenetic factors such as histone, heterochromatin protein 1 to this site, which initiate epigenetic programming. And this complex will further recruit other epigenetic factors such as histone methyltransferase to the site, which will further elaborate epigenetic programming by methylate, in this case, histone H3K9 residue. So this is what we initially discovered in Drosophila. And now in human and mammalian systems, we found that this complex can also recruit DNA methylase to the target site to achieve DNA methylation. So I'm happy to report that, up to now, our epigenetic study allowed us to discover a major mechanism in the germline cells that will recruit epigenetic factors to many sites in the genome to achieve epigenetic programming. And as you all know, such programming is very important for stem cell self-renewal, for germline development, for transposon silencing, and so on. So then you might wonder, what's the function of these Piwi and pRNA molecules when they're in the cytoplasm? So again, just to briefly review what we found in the past few years, we found that they actually play a very important role in mediating the regulation of transposons and pseudogenes towards protein coding genes. You all know that transposons are viewed as selfish parasites that invaded our genome, and traditionally, and still today, many textbooks regard them as a selfish element without any active function, except for potential advantage during evolution. Pseudogenes are viewed as the defunct carcass of our active genes on their way out during evolution. But we found that transposons and pseudogenes actually play a very important role in regulating the regular protein coding genes that we know very well. And this regulation actually is mediated by the Piwi pRNA complex. So back to the central dogma again. DNA for protein coding gene is transcribed into a pre-messenger RNA, which is then capped, tailed, and the intron spliced, become a mature messenger RNA and got exported into the cytoplasm. About 2 years ago, we discovered that about 50% of genes in our human genome actually contain transposon sequences in the 3' UTR of their messenger RNAs. That was a very surprising finding to us. And after that, we found that actually transposons are actually actively expressed at low level, at least in germ cells and primitive and some primitive stem cells. And the transposon expressed transcript will be processed into mature pRNAs and bound by Piwi proteins to form these Piwi pRNA complexes. These complexes can then, as you can imagine, beautifully target their corresponding or complementary transposon sequences that's hiding in the 3' UTR of mature messenger RNAs to regulate the expression of this messenger RNA. In addition, we found that the so-called pseudogenes, the dead copies of our active genes, are actually not quite dead. They also have a very important role. They are often expressed into messenger RNAs in antisense. And those antisense messenger RNAs will be processed by Piwi proteins to become pRNAs again and form complex with Piwi protein. And as you can imagine, this antisense RNA will be very capable of targeting the cognate active genes of the pseudogene. So now we know that gene regulation actually is not a linear way like what's stated by central dogma. It's very complicated, and pRNA and Piwi proteins play an essential role there. Likewise, if you are aware of new discoveries on some other non-coding RNAs, such as long non-coding RNAs, or called link RNA for short, even the expression of these thousands of link RNAs are regulated by pRNAs. And this regulation originates from transposons, because we found that many link RNAs, again, contain transposon sequences. So these sequences are like Trojan's horse inside their enemies. And the transposon-derived pRNA will, together with Piwi protein, bind to these target RNAs and regulate these target RNAs. So just to summarize this part of my talk as a background for the cancer side, again, if you liken our genome as the world, then the traditional genes that we study most of the time, about 23,000 of them, are part of the small world, the old world. And even in the old world, we often forget the presence of pseudogenes. In human, there are over 14,000 pseudogenes, and the transposons, over 1 million of transposons. And the pioneer work let us land ashore of a new world. Now the new world also contains another big class of non-coding RNAs called link RNAs. And pioneers actually serve a very important function to connect these two worlds. I've shown you very quickly in the schematic summary, pioneers can regulate the expression of traditional genes. Pioneer can regulate the expression of link RNA genes. Pioneer can regulate the expression of traditional genes through information from transposon. And even pseudogenes can use pioneer's pathway to regulate the expression of the cognate active genes. So now for this audience, you might be very curious about, does any of these basic discoveries have anything to do with the clinical side? We reasoned that because all these genes are so important for stem cell division, if you deplete any of these genes, the stem cell will differentiate into ordinary cell. We wondered if we overexpress these genes, will that cause malignant proliferation of stem cells? And will that be possibly a cause of cancer? So the question we really ask is Piwi pRNA function related to cancer? So a number of years ago, we took some human seminoma cancer patients, their testicular samples versus testicular samples from normal males and other kinds of testicular cancers derived from non-stem cell based malignancy. And we found that a human gene that we named Piwi, means human Piwi, is justly overexpressed in seminoma, which is known to be a stem cell derived testicular cancer. This, in fact, was the first gene to show such a high correlation to this type of cancer. So encouraged by that, in the past few years, we decided to look into other forms of cancer to see if any of these Piwi genes are overexpressed in these cancers. And we first took aim at the breast cancer, especially triple negative breast cancer for reasons you all know. And we first screened through 6 most representative lines of human breast cancer. And this is the normal human breast tissue, and this is the mouse one. And we looked for the expression of Piwi proteins or genes in these cancer tissues. There are 4 human genes encoding Piwi proteins. They are called Piwi like 1, 2, 3, and 4. And we found out all 4 are overexpressed, and especially Piwi4. In all 6 breast cancer cell lines, Piwi4 become detectably expressed. And in 5 out of 6 lines, Piwi4 become expressed at least 50 times higher than what's in the normal tissue. So encouraged by this cell line-based study, we decided to approach patients directly. And we randomly sampled 20 breast cancer patients for their pairwise tissue between the non-malignant breast tissue versus their own malignant breast tissue. And to our delight, we saw that 10 out of 20 patients indeed showed very strong overexpression of the Piwi genes. And to further confirm this correlation, we went to NCI database and to screen through over 1,000 cancer patients whose Piwi expression pattern was known. And indeed, we found that for those cancer patients with higher level of Piwi4 expression, they have much worse prognosis. So based on this very solid correlation, then we wanted to know what's the role of Piwi4 in breast cancer. Is this just a passive consequence of cancer development, or it could be an active role in promoting cancer formation and development? So in that, to address that question, we first did a cell-based experiment. This, you probably are very familiar, is a triple negative breast cancer cell line, highly aggressive. When we did the wound healing assay, normally if you remove this part of the breast cancer cells, just within 36 hours, the residual cells will highly proliferative and migrate into the central region to seal the void. However, if you just need to knock down the expression of Piwi4, then for a long time, much beyond actually 36 hours, these cancer cells fail to proliferate at high speed and they fail to migrate. And we've confirmed this assay by transfer assay, also indicate that PWL4 knockdown affects cancer cell proliferation and migration. Then we wondered through what kind of molecular mechanism PW4 achieve such amazing function. And it turned out PW4 is very important in promoting the epithelial to mesenchymal transition of these breast cancer cells. Because when we reduce the PW4 expression in these triple negative cancer cells, actually that alone can revert the cell fate from mesenchymal state back to epithelial state. So shown as an example here, E-cadherin is a typical epithelial marker and N-cadherin is a mesenchymal marker, and this is a loading control. The regular triple negative breast cancer cells are completely mesenchymalized. They express very high level N-cadherin, but no expression of E-cadherin is detectable. Now, if we just knock down PWL4 to 70% or a bit higher efficiency under 3 independent knockdown conditions, these cells actually now revert back to the epithelial fate. So that was very exciting to us because very few in the cancer literature, as you know better than me, people can see that kind of reversion. Then we say, why is PW so important in controlling the whole cell fate transformation? And we looked into the molecular pathway. Turned out PW4 promotes the TGF-beta and FGF-beta signaling pathways in cancer cells. So these signaling pathways are all highly expressed in breast cancer cells, but if you knock down PW expression, the expression is drastically compromised. In addition to this, we were surprised to find that PWL4 also inhibits the expression of MHC2 complex components in cancer cells. So this analysis allowed us to propose a model that actually PVL4 is a very upstream key regulator that promotes breast cancer oncogenesis. It does so by promoting these typical oncogenic signaling pathways activities, which promote the cancer cell survival, increase their proliferation, and cause them to have EMT. Meanwhile, the Piwi molecule will suppress the host immune system surveillance, allow this cancer cell to escape the immune surveillance. So this was the first somatic and major cancer type that we looked. Encouraged by that, we wondered whether any of the Piwi protein expression is correlated and possibly a cause of other major type of cancers. Then we looked into a second major type of somatic cancers, namely gastric cancers. And here, as you can see, you probably are very familiar with this histology. In the left panel, this is a histochemical staining with another Piwi protein called PVL1, now standing brown. You see in these adenocarcinoma sections from either the gastric body or the gastric cardia or the gastric entrant regions, there are robust overexpression of PVL1. And in chronic gastritis, which is a milder form pre-cancer condition, you see also atopic expression of PVL1, but at much lower level. PV1L1 expression is not there in the normal gastric tissues. When we quantify this expression level, it nicely correlates to the advancement of the gastric cancer stages. From stage 1 to 4, you see increased expression of PVL1, and also nicely correlate with the distant metastasis ability of these cancer cells. From no nodes to multiple distant nodes, you see again, these cells show higher level PV1 expression. And interestingly, its expression level is inversely correlated to the differentiation state of these cancer tissues or cells. So based on that, we decided that the PVL1 must likely also play a role in promoting gastric cancer. And to test that question, in addition to do the cell biology and the cell-based assays, as I showed you earlier, we also did the cancer xenographic transplant assay. So when we transplant these gastric cancer cells into nude mice, it grows robust tumors. And if you introduce the negative control for knockdown, it does not affect tumor growth. However, if you introduce siRNA that specifically knock down the expression of PV1 in these cancer tissues, you drastically reduce the gastric tumor formation. And when you looked into these tumor tissue sections by, for example, the cell proliferation markers such as PCNA, that's indicated in brown here, or Ki67, you see in the wild type situation, the cells are highly mitotic and prolific. In the control knockdown, you don't see any big difference. But in the PVL1 knockdown, you greatly reduced the proliferation of these cells. And that's a duplicate showing the same thing. So based on that, we wanted to know whether PINase play a role in these Piwi proteins function in cancers. We expected to see PINase and cancer-specific PINase to show up. But to our surprise, when we did deep sequencing for the small non-coding RNA population, we could not detect any PINase. We detected lots of microRNAs, which is normally a smaller population than PINase. But now, even with this big presence of microRNA population, we cannot see any, or if any, just very few PINase. That's very unmatching to the high level of Piwi protein expression and led us to conclude or to hypothesize that Piwi proteins function in cancer might be independent of PINase. To really demonstrate that's the case, we generated these mutant Piwi proteins by mutating 3 of its residue in the Piwi domain that you might remember from my introductory slides. That's possessing the RNA cleavage activity and PINase binding activity. So when you mutate these three residues, the resulting engineered mutant protein can no longer bind to pRNA. Then we wanted to see if these pRNA binding deficient mutant PWL1 proteins are still oncogenic, and turned out that's the case. So normally in these gastric cancer cells, if you do transfer assay for their migratory ability, they're highly migratory. Now, if you knock down the PW1 expression, you greatly reduced its migratory ability. But if you, in this knockdown cell, now reintroduce into these cells the mutant PW protein that cannot bind to pRNA, but that alone is sufficient to restore the migratory ability of these cells. So this analysis tells us that this PW function in gastric cancer, at least in gastric cancer, is actually pRNA independent. So our latest results showed that PW actually in gastric cancer, they do not interact with pRNA, but they interact with the nonsense decay-mediated RNA regulatory mechanism, NMD, to regulate the cancer genome or cancer transcriptome. What we think what's happening is that the PW protein, when it's highly expressed in these cancer cells, it directly targets tumor suppressor cells RNA and degrade these RNAs. So that allow the oncogenic RNAs to promote oncogenesis. And meanwhile, as you know, some tumor suppressor RNAs themselves will directly suppress cancer growth, and the suppression function is now knocked down because of PW overexpression. So we are very excited about these discoveries. In fact, this discovery started from Drosophila, and now we can all the way see the hope of approaching clinical site. Of course, I wanted to know whether, we can use this as a therapeutic target. And we have some preliminary results to show that's quite possible. So shown here is a new method that's being developed in my lab, is nanoparticles loaded with siRNAs against these proteins. In this case, it's against another RNA binding protein called familial that we also worked on and also has a tumor function, promoting function. You can inject these nanoparticles through tail vein into the mice that contains human cancer, in this case, colorectal cancer graft. And 28 days after that, after their growth, you do the injection, and after 4 times of injection, then you use a CT to visualize the tumor growth and the where microRNAs and siRNAs are localized. So we label this siRNA with Cy5 and tumor with luciferase. So by CT imaging, just 24 hours after the first injection, you can see these nanoparticles become highly enriched in the tumor cells. And then if you let these mice continue to grow, and you can see normally, if you just in the saline control or randomize the siRNA control, this grafted tumor tissue grow quite robustly in these new mice, and also will start to undergo metastasis, see other spots within 28 days. But if you do tail vein injection of these nanoparticles loaded with these particular proteins, RNAs, siRNA, you can really control the tumor growth. So, we really feel like our basic research allowed us to potentially find a completely novel oncogenic mechanism that's not specific to a particular type of cancer, but probably is to multiple type of cancers. Through our own data, we've shown that in breast cancer, in gastric cancer, in seminoma, and in prostate cancer, in skin cancer, in liver cancer, and in colorectal cancer, at least the subtypes of these cancers, they have high level of PW overexpression, and PW appear to be a driving mechanism for this type of cancers. So my dream is to use these PW proteins as target to treat cancer, to develop new treatment for cancer. Because PW proteins, as I mentioned earlier, they're only needed in the germline, and come to adult stage, they're only needed in the male germline, because female germline, namely ovary, does not have stem cells. Only spermatogonial stem cells are active here. So basically, you can completely knock out these proteins, the normal cell development will not be affected. We've done so, knock out entire PW family in mice. Mice are happily surviving. The only problem is the male mice, and we predict them. Male patient in human, they will temporarily lose fertility, but that easily can be solved by store the sperm right before the treatment. So this is my dream, and I hope with potential collaboration with all of you in the future, or some of you, we can take steps forwards, and hopefully there'll be a true new cancer treatment method will emerge from this. So I'd like to thank my lab members who did work. And since I'm running over, I'll stop here.
Well, Haifan, that was a fabulous body of work, and congratulations on all of it. And really, as you point out, it really does avail a whole new approach of cancer therapy. I know we're essentially out of time,
Right.
long, but let me just ask one question. Beyond potentially targeting through siRNA, are there other potential approaches to target these genes in terms of future therapeutics?
No, that's a very good question. Actually, the answer is yes, because these proteins, now the atomic structure have been resolved. So we know exactly how the active site looks like. And so we are also starting to use a small molecule-based screen to find these mimics of this active site binding substrate. And hopefully those will be specific to target and knock down these molecules too.
Well, that's exciting, and it sounds like a potentially new area for therapeutics. Well, I know we're out of time. I want to thank those who joined us, and thank Haifan and Grace for two really superb talks. Great work, and I thank everyone, and enjoy the rest of your day.
Well, thank you much for your invitation. Bye-bye.
Bye-bye.